Well, the aim of this channel is to quickly revise whatever we have learned in the today's class. We will discuss regularly each and every session of yakeen 2023. I have selected Tarun sir for subject biology. So uh, let's quickly revise whatever he had taught in the lecture number 1. Well, okay. Yeah, so this was the initial discussion. Let's directly move towards from where the actual study started. Well, okay. Now, what is cell? Cell is a kind of compartment which separate the outer and the inner environment, and wall or the membrane is made up of lipid, which is basically the phospholipid. It is water repellent. It's hydrophobic in nature. So the outer and the inner environment is separated by using the lipid or the phospholipid membrane. Okay. Chemical reaction, water, these are present inside the cell cytoplasm, and this membrane separates the two environment. That's what he meant by that's what is the definition of cell. Cell is a kind of compartment which separate the outer and the inner environment. All right. Cell membrane is present in all kind of cells, like eukaryote, whether it's eukaryote, prokaryote, archaebacteria, whatever the kind of organism it is. Every organism contains cell membrane, but the cell wall is there in case of cell wall is absent in case of animals. It is there in plants. It is seen in case of bacteria, but cell membrane is present in each and every living organism. Okay, now the question here is the one question. We will discuss each and every question, quick revision, okay, without wasting time. All statement 1 is like all living cells have cell membrane. Yes, all living cells have a cell membrane, each and every living cell. Okay, only dead cells lack cell membrane. Okay, like trachea, vessels, and the dead cells, like the only other example which we think of is any sclerenchymatous tissue, like not all, but mainly the sclerates and the those are the dead cells. All right. Requirements for any cells are like cell membrane, DNA which stores information for basic process, which contains hereditary information. The full form of DNA is like deoxyribonucleic acid. Organelles are present in each and every living organism, like that can be like membrane bound, or they can be without membrane. If they are membrane bound, then they are found in case of eukaryotes, and if they are membrane less, then they are seen in case of prokaryotes, like ribosome. It's there in both prokaryotes and eukaryotes. There is just the difference in their Sydberg units, right? Organelles are present, example ribosome. Then the movement of substance across membrane should be done so that oxygen, CO2, iron, the diffusion can take place. The food can give engulf, or the waste material can be excreted out. There should be some kind of movement in and out of the cell. So these are the few requirement of any cell. All right, so that's it. Then the difference between prokaryotic and eukaryotic cell. In case of prokaryotic cell, there is organelle. The organelles are present, but they are without membrane. Example, ribosome. Chemical reaction, all kind of chemical reaction takes place inside the cytoplasm. All the metabolic process takes place in the cytoplasm. DNA, here the DNA of prokaryote is known as nucleoid, but in case of eukaryote, it's not nucleoid. It's given the term chromatin or else chromosome, depending upon the condition, like on which phase of division it is. All right. So in case of prokaryote, all the DNA is there in the cytoplasm. All the chemical reactions are taking place inside the cytoplasm. But in case of eukaryotes, all the organelles can be membrane bound, and organelles can be without membrane. Without membrane organelles are found in case of without membrane, and this organelles are found in the cytoplasm. DNA is also present in the another chamber, and that chamber is known as nucleus. That is also again membrane bound structure. All right. Cell membrane is there. Membrane is of lipid. We just learned this in the beginning of the class, right? Well, in case of mitochondria, you will see the two membranes, like double membrane structures are also found, which separates the inner environment of mitochondria. Further into two compartments, right? Inner membrane and outer membrane. The ETS complex is there, etc. And even in case of chloroplast, you will see the two double membrane, means you will see the double membrane bound structure. All right. Reason why cell divide? Well, why cell divide? Cells divide just to pass the information to a daughter cell to next generation. Otherwise, if that didn't happen, every time cell has to evolve, then it will take millions of years. So cell has to divide to pass the hereditary information which it already contains. All right. So to pass the hereditary information, cell needs to divide. All right, let's move forward further. Now, what is the basic difference between DNA, chromatin, and chromosome? DNA is the linear, thread-like kind of structure which is double-stranded. It is a kind of nucleic acid, negatively charged. It has a phosphate ion, so that's the negatively charged. Chromatin, and the function of DNA is to store information for all process of cell, and the segment of DNA which contains the role, which actually performs the role, is that segment of DNA is known as gene. It is part of DNA which has some information. The part of DNA which undergoes, you can say, transcription process is known as gene. All right. It is a double-stranded structure. Then comes this chromatin. Chromatin is the condensed form of DNA, and chromosome is the further condensed form of chromatin. All right. Well, information is stored in gene. Gene is now here again is one question. Gene is made up of DNA, statement 1, and statement 2 is DNA is the functional segment of DNA. Functional segment is gene. Yes, that's very true. Gene is made up of DNA, and the functional segment of DNA is gene. Both the statements are very true. All right. Now let's move forward. Now here the two examples are kind of good questions. See, which of the following is nucleic acid? Well, the options are like SAL1, it is an enzyme. Hexokinase, again an enzyme. Tetracycline resistance gene, and Rubisco, which again an enzyme. Ribulose bisphosphate carboxylase oxygenase, right? So SAL1 is the enzyme. We know that enzymes are proteinaceous in nature except ribozyme, which is RNA, which is a nucleic acid. All right. If the option contain ribozyme, then we have to mark that as the answer, because they are asking us which of the following is nucleic acid. Then ribozyme is also kind of nucleic acid. RNA is a nucleic acid, right? So SAL1 is enzyme which is proteinaceous in nature, and hexokinase is also enzyme which is also proteinaceous in nature. Same goes with Rubisco. But tetracycline resistance is a gene, and we know that gene is a part of DNA, and we know that DNA is deoxyribonucleic acid. So that is also. So here our answer goes tetracycline resistance gene. It is a part of plasmid, which is a segment of DNA, extra chromosomal DNA. All right. Well, now here the next question given is, few examples are given like SAL1, ampicillin resistance gene, tetracycline resistance, Rubisco, and hexokinase. How many are nucleic acid? Of course, 2 of them are nucleic acid and other 3 are proteins. All right, that's why the answer is here 2. And if I add one more here, ribozyme, then the answer has to be 3. It depends like what the options are. All right. Now let's move towards the next slide. That is chromatin. Chromatin, DNA condensed form is chromatin, and in the chromatin you will see the histone, which are protein on which DNA is wrapped, and even non-histone proteins are also there. So basically chromatin is made up of DNA plus protein. All right. And why we need chromatin? Because why we need the condensed form of DNA? Because we need we want to store the huge length of DNA, which is around of 2.2 meter, in the very small nucleus, which is having the size of around 10 to the power minus 6 meter length. All right. So we need to store 2.2 meter length long DNA strand into 10 to the power minus 6 meter small nucleus. All right. Now the next thing is even the condensed form of chromatin is known as chromosome, which contains DNA plus protein plus some RNA as well. Chromatin is made up of, if the question comes and the options are like this, DNA only, DNA plus histones only, DNA plus protein, DNA plus non-histone, then our answer has to be DNA plus protein, because chromatin contains DNA also plus histone protein and as well as non-histone protein. So it has histone protein as well, and it has non-histone protein as well. So we have to mark DNA plus protein. All right. If we move forward, then it's like DNA is 2.2 meter. Why we need chromatin? Because 2.2 meter DNA has to be kept in a less space, and that's why DNA is getting condensed into chromatin. Well, why now your question will be like, why we want to convert, why we need to convert, why we convert chromatin further into chromosome. We convert it into chromosome just to when see, it's not that easy to divide chromatin into two equal parts. If the DNA is divided into chromosome, like it is a very condensed form of DNA, then only it will get divided in two proper equal halves, and that's why this DNA is converted into chromosome, so that it can easily divide. All right, so that's the motive actual of converting DNA into chromosome. All right, now here they are like explaining how this DNA gets divided, like how chromosome is like distributed among two daughter cells in a very intact form. These are the NCRT lines, we will read that as well. Chromatid, sister chromatid, these are the copy of each other, centromeres. They are just here is explanation is like, see, chromatin DNA replication took place. After DNA replication, we got two set of genome, and this two set of genome will now get distributed among equally distributed among two daughter cells. All right. Now if we talk about the condensation level, then the least condensed form of genetic material is DNA, and after that comes this compared to DNA, euchromatin is more condensed, and compared to euchromatin, heterochromatin is even more condensed, and the highly condensed form of DNA is chromosome. It is the most condensed form of DNA. So condensation increases from DNA to euchromatin to heterochromatin to chromosome. Okay, now when we require to open this chromatin during DNA replication and transcription, we need to decondense this chromatin to replicate the DNA strands. All right, now these are the few questions like this DOPA, like daily objective practice assignment. Let's quickly solve this question. Functional component of DNA is, again the answer has to be gene. Second question is cell membrane is present in each and every cell. As in the beginning of session we just concluded, this cell membrane is present in each and every cell, so answer has to be all. DNA in cytoplasm is off. In case see, DNA is in cytoplasm, it means like they are talking about prokaryotic cell, right? Otherwise, if the cell is eukaryotic, then the DNA is not there in cytoplasm, it's inside the nucleus. So we have to find out which cell is prokaryotic. RBC is again a eukaryotic, E. coli is prokaryotic, yeast is eukaryotic, which it belongs to the fungi kingdom. Amoeba, it's again a protist, so answer has to be E. coli because it's prokaryotic organism, so DNA is there in the cytoplasm. Organelles are present in all cells, not in some cells or not in bacterial cells only. Organelles are present in all cells like prokaryotic or eukaryotic. Alright, the next question is, in normal eukaryotic cell like muscle, DNA is present in the form of it is present in the form of chromatin. Generally it's present in the form of chromatin until and unless it's not dividing, but otherwise it's there in the form of chromatin only. Still we will tell you the answer at the end, like fifth A. We have just a doubt in this question, we will tell you it. Fifth, it has to be A. Alright, next is histones are present in where? Histones are present in chromatin and chromosome, so I will opt option number D, that is both B and C. DNA get decondensed for process, of course it has to be replication, transcription, so answer option D, both A and B. Most condensed form of DNA is, again answer has to be chromosome, that is option B. Question number 9, statement 1 is DNA is present in both prokaryotic and eukaryotic cell. Yes, that's true. Yes, gene is present in prokaryotic and eukaryotic cell. Yes, again both the statement 1 and 2 are correct. Euchromatin is transcriptionally active, and chromosome is transcriptionally active. No, chromosome is not transcriptionally active. And if you talk about euchromatin, so that is also not transcriptionally active. I will go with option number D. Still, let's see. Oh dear, they are like, okay, euchromatin is considered as transcriptionally active. Okay, though it's least condensed, even the least condensed form of chromatin is DNA, but I think euchromatin is not that condensed, so maybe it's transcriptionally active. So yes, the answer is A. Okay, and the answer of ninth one is C, that we also concluded that only. Then the doubt was there in the question number 5th, right? So let's just check. It's A only. Yeah, we even opted chromatin. Okay, okay, everything else is okay. Well, so here we finished off the revision of lecture number 1 of biology. We will discuss the all other sessions. We will revise all the 3 subjects together each and every single day. Alright, keep studying. Take care. Bye bye.